Já tá ok, já tá liberado.
Aperta a boca, senão no YouTube.
Good morning. We are going to start. I would like to invite Marcia Kobrusli to present our first lecture.
Thank you. Good morning. It's a pleasure to introduce Marluce da Cunha Montalvani. She's a graduate in biological science, medical modality, biomedicine. She has master's degree in biological science, biochemistry from the Institute of Chemistry of the University of São Paulo. She currently works as a researcher in higher level laboratory specialist at the Faculty of Medicine here with the Biotechnology and Innovation Center of the Technical Division of Support for Teaching, Research and Innovation at the University of São Paulo. And she works as a researcher associated with the NUCEL group in lines of research involving regenerative medicine, type 1 diabetes, isolation and encapsulation of pancreatic islets, as well as generation of endocrine pancreas spheroids and organoids. And she has experience in the areas of biochemistry, cellular and molecular biology, biotechnology, diabetes, and endocrine pancreas. Please, Marluce.
Here. Okay, good morning. It's good. You can hear me? Yeah. Okay, thank you so much. It's a pleasure to be here. Thank you for the invitation. My name is Marluce Mantovani, and I work here in School of Medicine of the University of São Paulo. And today we will talk a little bit about 3D culture and encapsulation of pancreatic islet transplantation. So, diabetes is a syndrome, a metabolic syndrome, and that affects the whole world. And it is expected that in 2045, about 629 million people are affected of diabetes. We have mainly type 1 and type 2 diabetes. And diabetes type 1, we have a pancreas failure of the produce of insulin, and the patient has, because of it, the hyperglycemia. Although this type 1 diabetes accommodates only 5 to 10% of the patients, these patients needs administration of exogenous insulin. In the severe cases, they need a whole organ transplantation, a pancreas whole organ transplantation. As an alternative treatment for this disease, we have the islet transplantation. So we have to remember that the pancreas has endocrine and exocrine functions, and the islets represents the endocrine pancreas, and it comprises almost 2% only of the whole pancreas. So in the islet transplantation, what we have is we take the pancreas from the donor, we take this pancreas to the lab, and in the lab, we isolate only the islets. And with these islets, we do the transplantation into the recipient. The transplantation is performed into the portal vein in the liver. So here in Brazil, our group was responsible for the first transplantation, islet transplantation, performed in 2002, and it was published only in 2004. But Despite this islet transplantation is a promised technique, we have some challenges that need to be overcome. So here we have the mainly challenges. So we have the IBMIR, the instant blood mediated inflammatory reaction after the insulin transplant. We have a poor revascularization of these islets after the transplant. We have the problems due to the immunosuppression of these patients, and we have the organ shortage. Today, in this workshop, I will focus in how we are working with our group to overcome these challenges in the organ shortage and immunosuppression. Last year, we published an article talking about the overcoming the organ shortage. So if you have interest, you can take a look at. So our group are interested in type 1 diabetes alternative therapy based on insulin producing cells. And now in the lab, I work with different lines of studies, and it comprises beta cell differentiation, islet isolation and transplantation, islet encapsulation, 3D culture, beta cell proliferation, and pancreatic decellularization. To start, to perform the islet transplantation, we need to isolate these islets. So here in this slide, I put some overview of the whole process. It takes about 10 to 50 hours to do it. So we receive the pancreas, we wave, clean and cannulate this pancreas, and we perform the organ perfusion and digestion using collagenases and proteases. The digest material is washed, and we put this material in equipment called Colby. Into this equipment, the islets and the acinar cells can be separated. And at the end, we have different portions, a portion that we have only islets, a portion that we have only acinar cells, and sometimes we have islets with acinar cells. With these islets, we can do the islet transplantation, or we can continue into the basic research. Trying to overcome a problem of organ shortage, we think about that we can proliferate these beta cells in the lab. So in a previous work of our laboratory, we showed that these islets, these 3D structures can be placed into adherent plastic culture. And these cells can come off the 3D structure and attach in the plastic and start to grow and proliferate. And we show that after they proliferate, they continue expressing insulin, secreting insulin, and progenitor markers. So it's a way to overcome the organ shortage. So nowadays we do it with human pancreatic islets and porcine pancreatic islets. So you can see at day 1, the islets attached to the plastic dish. And after 7 days, we only see single cells growing in 2D condition. And then we can expand and proliferate these cells, raising the beta cell mass. With this beta cell, we want to transplant these beta cells. But how we gonna do it? We are going doing single cells? No. Now, in this step, we want to do the reaggregate these cells before we transplant it. So the first technique that we used to do the 3D culture was using the nanoshuttle of grinding, grinder biotech, to do the 2D, pass these cells from 2D to 3D culture. So we start to verify different quantities of cells and establish different manners of do it. And we saw that after 24 hours, our cells comprise 3D structure without remaining cells, compact, and we can take it off the magnetic base, and this remains unsolved. So we did the histology of these 3D aggregates, and we saw that even with smaller clusters or larger clusters, the cells, they are compact and they form a layer and the outsides of the clusters. So here we can show that the cells are not only aggregate, but they are interacting and maybe becoming organoid because they can release insulin also. These 3D aggregates we use to recellularize our pancreatic scaffold. We did decellularization is another topic of these the studies that we have in the laboratory, and we intend to recellularize these scaffolds. So we use these 3D constructs to recellularize. And what is interesting to notice is that smaller clusters, they tend to attach to the acellular slices, and the cells start to come out the 3D structure and start to growing like we observed in 2D plastic surface. But the larger clusters don't. They innovate with the 3D extracellular matrix and interacts, but we don't have cells coming out of the 3D culture clusters. Okay, now we get this, we expand the 2D beta cell culture. We have the 3D for transplantation, but using this technique of nanoshuttle, we have just a little amount of 3D culture to do the transplantation. It's almost impossible because it's very expensive. So we try another techniques, and this I want to show you because you are trying to use this in your work. So here we use a plate called curarai to help to do this 3D structure. We use the hanging drops, the nanoshuttle, and we did use the agarose microwells. And hanging drop was a disaster. We didn't get any 3D structure. Curarai, we get 3D structure, but it was very difficult to manipulate. And nanoshuttle functioned very good, but it was very expensive. So we did these agarose microwells, and we get the same results that we get with nanoshuttle. So from here, we start to use agarose microwells. So after 24 hours, we get the 3D structures, clusters, well compacted. And these clusters, they are viable, and they are expressing insulin and pancreatic progenitors. We use this technique also not only with primary culture, but with Min6, that is a cell line, a mouse insulin cells, and we get the same results. We get round and homogeneous 3D structures. And for that, we use this to perform these agarose wells. We use this stamp. I get this stamp in a workshop that I was a long time ago. But these stamps has some issues. You have to hold this stamp until the agarose solidify. And we have in a plate of P12, only 2 or 3 wells, you get everything right to put the cells. So last year, a student from our laboratory improved this stamp. He made several modifications of this stamp. So now we are in the final, with the final stamp. And this stamp, he can be in the well by itself. So we don't have difference, methodological difference. And at the end, in one P6 well, we have 663 organoids clusters 3D constructs. So now that's how we are doing our 3D clusters using insulin producing cells from 2D culture, and after 48 hours, we get 3D cultures. This a view, microscope view of this well. So you can see the clusters are very homogeneous and concise and compact. So now we have this amount of cells, we have the 3D conformation of these cells, and we have to transplant it into diabetic patients. Trying to overcome the immunosuppression issue, we use a technique called encapsulation, cellular encapsulation. So nowadays, we know in the literature that the many biomaterials and delivery purposes are being used to improve the islet transplantation outcome. And in this context, we have the islet encapsulation. For the islet encapsulation, we have the microencapsulation, microencapsulation, and nanoencapsulation. In the lab, we start to use the microencapsulation to perform microcapsules. In the literature, there are many of products that can be used to generate the capsules. Our biomaterial has alginate as a basic material. And what we want using the cell encapsulation is that the insulin producing cell is inside the capsule. This capsule can permit the glucose, nutrients, and oxygen get in, but don't, and the insulin and the another metabolic release of the cell come out, but the immune system can attach, can enter in the capsule. So using this technique, we can avoid the immunosuppression in the islet transplantation. In our lab, we use alginate in the base of our biomaterial, and we do the encapsulation using a simple, more or less simple technique where we have airflow in the top and in the middle of the solution. We have our biomaterial, our cells, and another additives that we want to put into the capsule formation. We have the drops of these biomaterial and cells, and we have airflow. These drops fall in a gelish solution, and then we have the formation of the capsule outside the cells. In a previous work of our group, we showed that a biomaterial that we used were able to protect the islets, and these islets were transplanted into mice, diabetic mice, and these diabetic mice maintain normal glycemia until almost 1 year. And after almost 1 year, these capsules with cells were removed, and the mice become again diabetic. So we improve these capsules, and this is another work of our group showing that the incorporation of extracellular matrix component called laminina and polylaminina improve the results that we have with our previous biomaterial. So here are the capsules that we work in our laboratory. Biodretin and Bioprotect has patents, are laboratory patents, and we are trying to improve a little bit more these capsules to improve the islet transplantation outcome. So now we are in collaboration with Professor Flávio Galvão and Giovanna to try to perform this pancreatic islet transplantation microencapsulated into pigs. So we are in the first step of this work. We are trying to standardize the pancreatectomy in pigs. And after this pancreatectomy, the pig is, we expect they become diabetic. And after this removal, we intend to transplant the 3D insulin-producing cells, microencapsulated, and we hope this can reverse diabetes. Moreover, we are always trying to improve these capsules. Yesterday, this work was accepted for publication, where we decellularized the pancreatic, and we get a cellular pancreatic scaffold, and these cellular pancreatic scaffold are made of extracellular matrix. So how we saw improvement using only laminin. The idea now is trying to incorporate the whole extracellular matrix with all the components after the cellularization into the capsules. So we start with this work using different concentrations of human decellularized extracellular matrix and porcine decellularized extracellular matrix incorporated into the capsules. We are in the initial, getting our initial results. But as you can see, the surface of these capsules are very similar with our control. That is our best condition of the capsule that we use for transplant. So which always encapsulate the islets, the porcine islets, and there are no cytotoxicity shows in using these extracellular matrix in different conditions. Now we are continuing with these experiments, but we are very happy with the results so far. Yes, last year I was in the University of Groningen, and we started a collaboration with Professor Marina Trombetta Lima and Professor Paul de Vos. And with this collaboration, we intend to analyze if the extracellular matrix in capsules may protect the human islets from stimulus stressors. So we get the preliminary results showing that maybe our hypothesis is correct. And today this work is being presented by me in an international congress. So the summary of everything that I showed to you today was that we can isolate pancreatic islets, human and porcine, using this methodology that we established in our laboratory. This 2D culture can be done, enabling insulin releasing for a long time period of these human and porcine pancreatic islets. We are able to do 3D culture using nanoshuttle from Greiner, but we are able to do 3D culture using agarose microwell as well. This 3D culture can be used to recellularize acellular pancreatic scaffolds, slices, maintaining functionality and viability of these cells. We developed a stamp for agarose microwells where we can get 663 microwells per P6 well. We can do the cellular encapsulation of these cells, avoiding the immune response. We are establishing a surgical diabetic pig model with Professor Flávio Galvão. We are trying to transplant 3D insulin-producing cell encapsulated using this model, hoping that it reverses diabetes. And we're trying to improve these capsules using the cellularized pancreatic extracellular matrix. So thank you very much. I want to thank for the organization committee and all the agents, the Professor Meire Sogayar, her laboratory, my colleagues, especially Henrique, Rafael, Amanda, Professor Roger, that's head of DataPep, that I work, Professor Galvão and Giovanna, Professor Marina, her students, and Professor Paul de Vos with her student. Thank you very much.
Thank you very much, Marluz, for this brilliant presentation. And we'll keep forward with our workshop. Introducing you the next presenter, Dr. André Barateiro. He is postdoctoral research at the Unit of Drug Discovery Department of the Parasitology in the Institute of Biomedical Science from the University of São Paulo. Dr. André Barateiro actually is our collaborator in one research. He was graduated from the University of in the Bachelor of Master in Genetic and Molecular Biology biology at the Faculty of Science, University of Lisbon. And actually, he is performing his postdoctoral in the with Dr. Wangler, that is a very good research, and the chief of the parasitology in the Biomedical Institute from the USP. And he's doing his PhD in collaboration with us in the hepatocyte culture. Today, he is performing his presentation entitled 2D culture of primary human hepatocytes to start the liver stage of the malaria parasites. And thank you very much, Dr. Andre, for your participation in our workshop.
Can you hear me?
Yes.
I cannot share. I'm not allowed to share the screen. Ah, now I am. Okay.
André, você pode conectar agora.
All good. Can you see the slide? Is everything okay? Can I start?
Yes, yes.
So good morning, good afternoon, good evening, depending on where you are. Thank you for the introduction and for the opportunity to be here with you all. I just want to say at first that this, I feel like a bit of an outsider because I've never done any research on pancreatic tissue nor hepatic tissue, and I also don't have fancy data to show. Rather, I will tell you a little bit about the story of how I got here and how I am trying to implement, in collaboration with people from the Faculty of Medicine, a 2D culture of primary human hepatocytes to study the liver stages of the malaria parasite. So in the beginning, I finished my PhD, and the only thing I knew was that I wanted to keep on studying malaria, but I needed to find some knowledge gaps that could render a potential postdoc in the future. So the first thing I went to check was what were those knowledge gaps. So what we knew and what we know is that malaria is this disease caused by a protozoan parasite from the genus Plasmodium that is injected by an Anopheline mosquito in humans. And this parasite, like in the beginning, is fragile, and he fastly travels to the liver where he chooses an hepatocyte, and that's the hepatocyte that will help him develop into another form called the merozoites that will be released in the bloodstream. And this is the point where the disease gets to be noticed, where the symptoms start to happen, and that can lead to some fatal outcomes. After this, the parasite produces the sexual form called gametocyte that will be later sucked by a new mosquito that will then help him to fulfill the cycle and then be transmitted to a new human again. What we also know is that the disease still represents a big burden to the world because every year there has been reports of roughly 250 million cases, most of them in Africa, that normally lead to approximately 500,000 deaths every year. And we also know that despite the fact that, okay, let me get a pointer here, sorry. Okay. Despite that the fact that there are 5 known parasites that actually infect humans, most of the cases are caused by Plasmodium falciparum and Vivax. And this disease is roughly distributed all around the low and mid-income countries spread across the world. And also is a disease that is still present in Brazil. But in Brazil, we also know that the proportion of falciparum Vivax cases, it's inverted. So there are more Vivax cases. And this should be encouraged because the Vivax normally don't lead to fatal outcomes that often, but he has unique features that make him a challenge for elimination. So we know that this parasite in particular creates low density and asymptomatic infections that are normally undetected. It has a high production of gametocytes early on, so they can be spread before the symptoms to occur. And it forms this very resilient and undetectable form in the liver called the hypnozoite that is known to be roughly responsible for 85% of the cases reported of this species. It is known that the drugs that are used to kill it often depend on the CYP2D6 enzyme status of the patient, because the drug that is mostly used, Primakin, depends on this activity to be active and kill the parasite in the liver, and also depends on the G6PD status of the patient. If you have low expression of G6PD, you tend to get hemolytic stress after taking the drug, which is not that good because the patient can have a very bad outcome after taking this medicine. And so far, there are no reliable biological, neither serological markers to diagnose these carriers of this dormant form. So with this research of the literature, I found some opening for innovation at this point. And I also realized that nobody knows what triggers activation and relapses. Relapses meaning that when the parasite gets released once again in the bloodstream, causing disease. So by finding this, we identified this opening for innovation. So I decided to pursue this in my postdoc. But one thing I needed that is not something that is that often done in Brazil is a model. So a cellular model to study this. So I have been pursuing this for the last 1 year. And I'm going to tell you some key things that I found important to consider when trying to implement this kind of model. So we need to know the scientific environment and the resources and infrastructure that we have in order to get this models going. And we also need to be aware of the characteristics of the parasites and the cells. So we need to question ourselves of what do we want. So we want a parasite source. In this case, we are going to use Plasmodium vivax to get those hypnozoites, and we need to decide which host cell do we want. So there are a lot of, so we need to use the most abundant cell in the liver, which is the hepatocyte. But there are several ways that you can study this hepatic cells. So you can use hepatomal cell lines such as FG2 and HCO4 cells, hepatocyte derived from pluripotent stem cells, or you can also use primary hepatocytes. So, but we need to consider the following. So in order to see hypnozoites, we need to be able to maintain these cells for a long term in culture, because the hepatocytes normally appear in vitro around day 8, day 10, and they only reactivate after day 15, more or less. So we need to keep cells for a long period of time. We need to make sure that these cells express drug metabolism related enzymes, such as CYP2D6, if you want to test some drugs that depend on those enzymes to be active. And we also need to consider that the expression of invasion receptors. So this can drastically affect infection rates. And depending on the parasite, we need to make sure that the proper receptor is there. So falciparum depends on the expression of CD81, while Vivax depends on the expression of ephedrine receptor A2 and SRB1, which, as far as I remember, is also an important receptor for hepatitis virus. But so there are experts around, so they will be able to correct me afterwards if I said something wrong, okay? So let's compare these cells. So in terms of long-term culture, so hepatomal cell lines are impossible to keep for long periods of time in culture because they tend to overgrow, and diluting them afterwards might disrupt the development of hypnozoites. Say they are not a good candidate. Like long-term culture of hepatocyte derived from pluripotent stem cells, to my knowledge, was not reported so far. And primary hepatocytes are known to be able to be maintained for long periods of time in culture, but this is also depending on the system. We shall talk about that later on. These two first types of cells regarding drug metabolism enzymes, so they present low to absent expression. while primary hepatocytes have the expression of these important enzymes, but this is also donor dependent. Regarding invasion receptors, depending on the cell line, so the receptors are not expressed. So some of them express one of the receptors, others express others. In these hepatocytes derived from stem cells, the expression of invasion receptors has been reported, but they lead to low invasion rates, while primary hepatocytes tend to have a variable expression of these invasion receptors, which is also donor dependent. But like if we compare all of them for what we want, primary hepatocytes are definitely the best cells to choose to get for us to be able to answer the questions we want. So after choosing the cells and the parasite, I needed to go after potential collaborators that would allow me access to this. So we have now ongoing collaboration with Dr. Maísa Araújo and Dr. Jansen Medeiros at Fiocruz Rondônia. So they are able to produce Vivax sporozoites in a stable manner. So they will be the source of it, and the primary hepatocytes. So I will have access to them with the help, only because of the help of this group of people that I have here. So Dr. Flávio Galvão, Dr. Márcia, Dr. Paulo Herman, Dr. André Irayama, Dr. Venâncio Alves, and Dr. Evandro de Melo will be actively participating in this process. So, and I'm going to tell you, so let me just take the opportunity to acknowledge them and thank them all for their help so far. So they will help to solve this problem. So how do we get these cells? This is the tricky part. So let me apologize now for, so I'm not a clinician. I don't know much about this. So any misuse of terms here. So I apologize for it because it's completely out of the scope of what I've been doing so far. I will try not to mess that much. But like we decided that patients with hepatic tumors that will be submitted to liver resections will be the best donor candidates. So what we plan to do, so we are now at a point that we want to get ethical clearance for this. We want to recruit these patients, okay? And if they accept, they will willingly give us their liver resections that otherwise would be sent to the trash for us to isolate the hepatocyte. So the idea is so imagine that we get like a piece of liver like this, just as an example. There must be a lot of things wrong with this drawing, but just try to keep up with it, please. So assuming that we get a tumor here, the idea is to have it taken for pathology examination, and then to get the healthier part of the liver. Like we will try to get the glycan capsule intact because we need to do a perfusion afterwards. The blood vessels need to be intact and preserved. So we will have a single point cutting more or less at this point. So we can do a nice perfusion of the tissue afterwards with digestive enzymes, okay? So after this being done by the pathology team, we will get that healthy part of the tissue that otherwise would be trashed and do what we know as a two-step collagenase perfusion protocol. So the idea is then to perfuse the liver in 2 steps with 2 buffers, one of them without calcium and with ADTA, to block the desmosomes that keep cells together and facilitate their dissociation afterwards, and afterwards perfuse the system with a buffer containing calcium and collagenase that will destroy the extracellular matrix of the tissue, make it easier to release the cells for us to purify afterwards. So this is basically like what a perfusion system looks like. So you need a peristaltic pump to pump liquid through a tubing system to a water bath that will warm the medium and then channel the liquid inside the liver. So this is a liver from a monkey. So I went to Rijswijk, to the Biomedical Primate Research Center, to learn this technique last December. And this was where the picture was taken. So after this, and after passing the collagenase through the tissue, we will be able to mince and smash the tissue easily and release the hepatocytes in another type of medium, which we can pass through a strainer to just get rid of the big chunks that were not smashed. This can be washed and then put it in a percol to do a gradient centrifugation. With this, we will be able to separate easily the hepatocytes that are way denser than other cells. And even if we want, we can use this method to isolate other types of cells for other kinds of research. But we want hepatocytes, so after this step, you can wash them, count them, check for viability, and use them for experiments. Attention, it's important to consider that you can also cryopreserve these hepatocytes with specific techniques. This is important. Why? Because once you identify a good donor, you will be able to use it for several different experiments, reducing the variability between results. So having the primary hepatocytes and sporozoites, we need to consider the way of cultivating these cells. Why is this important? Because we need to ensure that it seems that there is a correlation between the long-term maintenance of these cells in culture and their maintenance of hepatic phenotype. So we need to ensure that these cells look like hepatocytes in the long run. And it seems that, so maybe Dr. Bassett and Dr. Michaelidis that have more experience on this matter can comment on my generalized assumption of this, that confinement and support seems to be correlated with longevity in vitro. So if you, for instance, use micropattern co-cultures that have also non-parenchymal cells on the side, like you can see here, like fibroblasts, or even if you have cultured in 384 well plates, or even micro wells created within macro wells, like you see here, you can have this polygonal shape very well defined, and this is correlated with the longevity of the cells in vitro. Because if you don't have this, like hepatocytes become not so polygonal, and they lose their hepatic characteristics in vitro in the long run. So considering this, we need to choose a model of a way of culturing them that ensures their long-term maintenance of hepatic phenotype. And once we get that, we need to characterize it. So a good culture of hepatocytes, like in the long term, can be evaluated by microscopy, like in a bright field view of the wells. So you can do a morphologic evaluation. You can check if these hepatocytes keep on producing albumin across time. You can also check if there is the formation of bile canuliculi, which is a proxy for transport. And you can also check if mitochondrial activity is on throughout time, which means that the cells are alive and metabolically active. So you need to do all of this and even check for the expression of those drug metabolism enzymes that I mentioned before, and the expression of those relevant invasion receptors that without it, we cannot achieve this. And what is this? So liver stage infections. So once we get a good donor and a good culture system, we need to check if we can infect them and if the parasites can develop inside the hepatocytes. So what we have here is, just to mention that the readout of this kind of approach is mostly microscopy, because the only way of distinguish between an hypnozoite and a schizant, which is the developing form that is not dormant, that is inside the hepatocyte and afterwards gets released in the bloodstream, it's via size discrimination using microscopy. So you can get constitutively expressed markers such as this US4 protein that is expressed both by hypnozoites and schizants, and see that they are completely different in size from each other. So we can use this to identify one and another and do whatever we want to do and to discriminate them and then study them accordingly. And by knowing this, we need to consider so this is also important for the type of plates we want to use, because, for instance, knowing that this kind of plates and culture systems is definitely correlated with the throughput capacity of an assay. So this would go from medium to high throughput, and this would go from, it's a low throughput culture way of having these models. One needs to consider that at this point the infrastructure that one has. So if we go for 96 well plates or 8 well chambers, for instance, we need to have a regular or inverted fluorescence microscope. While if we go to a 384 well plate, for instance, we would need to have to take pictures of these parasites, high content imaging systems such as the operetta. So this was a tricky one to solve because not everybody has this kind of systems. And the idea of this project was also to extend the model to other institutes across Brazil. So if I want to run this model in Rondônia, for instance, I need to consider that they probably will not have a high content imaging platform there. So to solve this, we decided to try to, instead of using like a co-culture system that depends on the micro patterning of the well. So in this case, when you have a micro-pattern co-culture, you basically tell where the hepatocytes are seeded, and the outer area of that aisle will be crowded of fibroblasts. But in a self-assembly co-culture system, so this was already used to culture, to infect hepatocytes with hepatitis virus, one can get a long-term culture of hepatocytes supported by mouse fibroblasts in this case, simply by allowing them to self-assembly inside a well. So you basically throw both cells together and they rearrange in space, and this is enough to get hepatocytes to reach like long periods of culture in a healthy way, okay? And it's important to mention that this was never used in experiments that aim to study hypnosooids. So this is a point of innovation that we are suggesting with the project. So overall, what we plan to do is, like, this is more or less the experimental design. So this is something that you need to have in order to culture hepatocytes. So you need to pre-coat wells with a collagen matrix. Afterwards, you allow the hepatocytes to be seeded, and the day after, you throw the supportive fibroblast there. So in this case, you can use like a very well-known fibroblast line that is enough to support the growth of the hepatocytes and keep them for long periods in culture. After this, you evaluate the hepatic phenotype of the culture, and then once you get everything going, you can infect the culture with sporozooids. And this should be enough to allow us to get all the liver stages of the parasite in culture and not only hypnosooids. So with this running, you can apply this to the most diverse, to try to answer the most diverse type of questions. So just to give you some example of the applications that we can have from having a model like this in Brazil. So we can test new drugs. So just as an example, so Primakin is a drug that is known to kill both schizons and hypnosooids in the liver. So it's given to people that have been infected with Vivax, while Atovaquone is a drug that is given in a prophylactic regimen. So it doesn't kill hypnosooids, but it kills liver stage schizons. So since we are able to discriminate between bigger schizons and small hypnosooids, we can basically do get drug response curves by just analyzing the different sizes of the parasites that we get in culture. Okay, so here you can see the applications of this. So you can see that Primakin, in a dose-dependent manner, is capable of killing both small, which should be hypnosooids, and large schizons, parasite forms, while Atovaquone is only able to kill the larger forms of the parasite while not being effective in killing the smaller forms. as expected. So then you can use this in the most diverse types of drugs and test them by using this model. You can also do more basic kind of research. You can simply try to get hypnozoite-enriched cultures and compare them to a mixed culture of hypnozoites and schizants. So in this case, some research did a comparison of their RNA expression profile, and they were able to see that the transcription profile of hypnozoites is completely different from a mixed culture. And back then, it was thought that these hypnozoites were dormant and inactive, but now, and this kind of research allowed to show that these hypnozoites are actually not dormant. They might be latent, but they have some metabolic activity going on. And from this, we can have these kind of notions and go to more other types of experimental approaches. So here, a group of researchers was able to show that the hypnozoites inside hepatocytes have these protrusions, so these tubules that come after the main core of the parasite, and that this parasite in this stage and these tubules is able to recruit a protein called aquaporin. So this is actually the human aquaporin. So it means that the parasite is recruiting the host proteins, and it's thought that this protein is being used as a scavenging strategy. So parasite probably uses it to get nutrients from the cytoplasm of the host, and with this kind of model, so these researchers were able to show a very nice way in which the Plasmodium hypnozoites parasite its host. So, but you can do all sorts of different kinds of things. You can even use this model for studying hepatitis, other types of stuff that probably might not be related with parasitic diseases at all. So yeah, having a model like this going on in Sao Paulo can be very important to galvanize any type of research that we want in the future. So this is just like what I wanted to tell you today. I don't have results, like I said, because my project just got approved in March. So my fellowship will only start in August, but I want to just take the opportunity to acknowledge and thank FAPESP for once again trusting these kind of projects that I think will be very important for our community. So let me thank like all the people involved so far. So without them, I wouldn't be able to be running this. So I want to thank my supervisor mostly because he trusted me to conduct this project. I want to thank my collaborators in Rondonia. Also, these collaborators that I didn't mention, Dr. Stephen Maher in the University of Georgia and Dr. Noah Sadler in Seattle Children's Hospital. They are important in giving me some help because they know how to implement these kind of models. They have been doing it outside Brazil, in this case, in the States. So they know how to deal with this kind of stuff. Dr. Anne-Marie Zeman and Dr. Clement Cocken, which received me in Reichswijk and taught me how to isolate primary hepatocytes from primate livers. And all the people that have been involved in the implementation of this project at the Faculty of Medicine. So once again, thank you, Professor Flávio, Dr. Márcia, Dr. Paulo Herman, Dr. André Irayama, Dr. Venancio Alves, and Dr. Evandro de Melo. Thank you for the organization for inviting me. And I think that's it. Thank you for listening. And I'll be open to answer the questions that you might have.
Thank you very much, André, for this brilliant presentation. Very nice. And we're going to make a break. 30 minutes, 10, sorry. No final. Quer fazer algum?
André, are you there? He's
Yes,
not
I
there.
am.
Oh,
No, I am.
hi. Hi, André.
Hello.
My name is Mary Cleide Sogayer from the Chemistry Institute of the University of Sao Paulo, currently at the medical school. I just want to ask you about your co-culture in which you use fibroblasts to improve the hepatocytes' survival and functioning. It's a very good idea. But on the other hand, I noticed that you use murine 3T3 cells as fibroblasts, which are murine cells, and you're culturing human hepatocytes. So one suggestion would be to change to human fibroblasts. And another suggestion would be to use no cells at all, but the conditioned media from fibroblasts that should contain small vesicles, exosomes, that would probably do the same benefit as the whole cells. And I want to congratulate you on the work, because culturing hepatocytes is really crucial to maybe do the so-called temporary transplant, since many patients die waiting for a liver. It would be really important to have hepatocytes culture to allow them to live at least until they get a donor, like 2-3 weeks. But please consider the idea of using either cells, human fibroblasts, or conditioned media from these cells. That's it.
Thank you very much for the suggestion. So the point is, like, these things, like, I feel like I'm not being that innovative. I know that I'm trying to be innovative in the questions I'm bringing in relation to malaria specifically, but these things have been done for the past 10 years, like, quite frequently. The reason why they use murine fibroblasts is because this is actually a cell line. It's not primary fibroblasts, so which is way easier to maintain in culture than primary isolates, as you know for sure. If you have, because this would imply to separate also primary fibroblasts in an isolation protocol as well. So if I don't know this, I really don't know if there is like a fibroblast cell line.
Yes, there
of
is.
human fibroblasts. There is. Can you give me the name,
There
because
is.
this is important to
Yes.
consider?
It's called WI38. It's a
W?
normal human fibroblast cell line.
WR.
WI is an ISIS. WI38. There's also another cell line called VA13, which is the same WI38 cell line transformed by a virus. I don't know which one, SV40 or some, or even not the virus, but the oncogene that allows immortalization. These are immortalized fibroblasts, human fibroblast cells, totally immortalized. The WI38 cells survive for at least 50 generations in culture, and you can prolong their lifetime in culture by changing the medium, by supplementing the medium with different growth factors. But if you want a really permanent cell line, then you would like to use VA13. Look into them. The American Type Culture Collection has both of these cell lines. You can look at them.
Great, thank you. Let me just comment on what you said, like, of not using supportive cells at all. Actually, that's a tricky one, because when I came up with this, I wanted to do like a thing that could be used also by the collaborators in Rondônia. So I know that they don't have an operetta, so they cannot run this in a 384 well plate. This is what I plan to do in São Paulo because we have an operetta at ICB. So like you said, like using no supportive cells at all would be the best case scenario. But I'm also considering the possibility of running this in a 96 format or a 8-well chamber format that could be also used in other states that don't have such a fancy kind of infrastructure. Yeah. Thank you very much for the comments and for the help.
Thank you very much, Professor Mari. And okay, we're gonna have our break. 10 o'clock here again, okay? We have a cafeteria in the basement, so you can go there to have some coffee. Thank you. See you in a little bit.
So let's continue. We now introduce Dr. Glauco Souza. He's the Director of Global Business Development of Innovation, 3D Cell Culture at Greiner by One, and former president and co-founder of Nano 3D Bioscience Incorporation. He's one of the creators of magnetic 3D cell culture, including magnetic 3D bioprinting. Dr. Souza is also an adjunct assistant professor at the University of Texas Health Science Center at Houston. His work has been reported in several high-impact scientific journals, and now Glauco's mission is to transform 3D magnetic cell culture into a routine laboratory tool that will measurably improve the process of drug discovery, cancer research, personalized medicine, and regenerative medicine. And his work was recently selected for the short list of the 2017 Lush Prize for Outstanding Research Producing an Effective Non-Animal Safety Test. And now, please, Dr. Glauco.
Thank you very much. I'm happy to know that I can give this in English too, because usually I'll give the presentation in English, but if you wanted to ask question in Portuguese, you're more than welcome. I'll answer it. And I'm sorry I cannot be there this year live, but I'm very happy to be part of it. It's a great venue. And I'll be talking about magnetic 3D cell culture. And since you guys have been hearing a lot about 3D cell culture and the whys of 3D cell culture, I'm going to try to focus on practical aspects of 3D cell culture. And of course, the practical aspects have to be validated with real data. So I'm going to give you a tour of a lot of the results we have. First, since many of you are working on 3D cell culture, and I want to sort of open this and let you guys know, if you have a good protocol and you want to publish on a great journal, SLS Discovery has a special issue for 3D cell culture. I'm one of the editors, and we're accepting papers. And again, these are protocols for 3D cell culture. And if you have any questions, you're more than welcome to reach out to me to see how we can make it fit in the issue if it's appropriate. And as I said, I'm one of the editors. We had 2 years ago, we had another issue, which was get some great papers and some from Brazil as well. And next week, it's a little bit of a short notice, but for the following year, this is a great symposium. It's called BB3D from SLAS. And it's a really great venue to learn and share results as far as 3D cell culture. Will be next year at Scripps, Florida. And I'll be on I'm leading the enabling technologies session, and that's what I'm going to talk about here a lot, which is enabling tools for 3D cell culture. So I'm not going to get into the why 3D cell culture. I'm assuming that you have heard a lot why we're moving to 3D cell culture, but I always like to show this result. And this is it's a little bit of a history of where I started with 3D cell culture. So we started in 2008. In 2008 was there was already work in 3D cell culture, but there was, as we say, a lot of hand waving on that 3D cell culture is a lot more like in vivo. 3D cell culture is different from 2D. And 3D cell culture is better than 2D cell culture. So, and this was the result that we published in Nature Nanotech. And I like this image because it clearly shows you don't have to be an expert. You don't even have to be a scientist to look at this images here, this column and this column, and see they're different, right? So this is a glioblastoma, paraffin-embedded glioblastoma culture. In this case, is a PDX. On the right is a 2D cell culture of the same cell types. I shouldn't say PDX, it's a xenograft because these are cell lines. So it's LN229. And we stand for encadherin. Encadherin is a protein that if you're doing 3D cell culture, that usually comes up. It plays a major role in the assembling the tissue. So we're just showing here that the red is the immunofluorescence staining for encadherin. And you can see 2D is certainly different from in vivo. So then we did our culture using magnetic 3D cell culture, and that's the result we got. So I think you don't have to be an expert. And if you see the tissue in 3D, what does the tissue look like? The staining, the encadherin distribution. It looks like in vivo. So, and this is probably one of the reasons we got this paper in Nature Nanotech. And that was very early on. And that's what we needed to show that, 3D cell culture, it is closer than in vivo biology, and I will show more results as we go. So where did technology come from? The technology came from collaboration between MD Anderson. I was at MD Anderson at the time, 2 other friends from Rice University. We came together with this project. We got these great results. We published, and then we formed a company. We published, we patented, and we formed a company called Nano3D Biosciences. So I moved to Nano3D. I led Nano3D Biosciences for 10 years. And then as of 2018, Griner acquired the technology. So, which is why I'm at Griner now. So it really started from basic academic work. And as I said, I'm going to focus a lot of the practical aspects of 3D cell culture. And I do like this quote: everything should be made as simple as possible, but no simpler. And when you go to 3D cell culture, let's think that 2D cell culture is the simple way of doing it, right? And when you move to 3D cell culture, you're adding a dimension, you're adding a dimensionality, basically, right? And you are making a more complex system. So it is going to be more complicated. So we have to accept that 2D and 3D are different, and 3D is more complicated, okay? So, but a lot of times we make it more complicated than needs to be. And it's funny, I'm guilty of that too. We always want to do try different things, and all of a sudden it's more complicated than it has to be. Then you also make the mistake you try to go back. You got to strip things off of the process, and you go too far again to make it very simple, and then you're back where you started. So you have to accept some level of complexity. That's why, made as simple as possible, but no simpler. So, and from the practical aspects, one thing that you learn when you go from 2D cell culture to 3D cell culture, it's true, you do appreciate the value in how easy it makes our lives when we have cells stuck to plastic. When cells are attached to plastic or glass, it's so much easier because the simple task of changing media, right? when we're culturing cells, we need to change media. You aspirate your media out, you add your media, and cells are attached, right? That's easy. Immunofluorescence, the cells are attached. You add your blocking, you do your PBS wash, you add your antibodies, and the cells don't go anywhere for the most part, right? So, but if you go to 3D cell culture, you lose that. It's not easy anymore. Changing media, it's not simple anymore because your cells are just floating usually. And then you lose your cells most of the time that you do the washes and manipulation. Immunohistochemistry, it's not easy. If you're using a gel, especially immunohistochemistry, you have a gel there. How do you get your antibody, your solution? How do you wash or your cells when they're stuck into the gel. Then you have to remove the gel, and that's usually complicated because you have to digest that gel if it's a protein gel, protein-based gel. And that will also change your cells, because when you're digesting the gels from the protein, you're digesting the proteins on your culture. So it is more complicated. So one of the reasons we came up with magnetic 3D cell culture is to make it simpler and to make it closer to 2D cell culture. And how do we do that? We magnetize the cells. So we use the magnetic field as a surrogate for cell attachment. So we have this nanoparticle, we add to the cells. It's gold iron oxide nanoparticles cross-linked with polylysine, and the nanoparticles just stick to the cells electrostatically. And once they stick to the cells, you can use the exactly workflow that you do with your monolayer culture. The only difference is you have to put the magnet. You either put the magnet on top where the cells actually levitate and form the 3D cell culture, levitating. And this is what we call the low throughput. We have this in 6 and 24 wells. And we have the high throughput or all throughputs, which is you can do from 6 all the way to 1,536 micro-well format. But this time we use a cell repellent surface, so a surface that the cells won't attach to it, and you can put a magnet on the bottom, and you can really miniaturize your culture and make it very easy to manipulate at the bottom. And this is also allows us to do the high throughput. So the magnetic field here is just a way of making the culture process easier. And I like to say this, emphasize, and it's true. There are many steps when you're doing 3D cell culture that are difficult. And you may be able different tools, but even if you're using different tools, if you magnetize your cells and use the magnets, you can make those challenging steps easier. So, and even if you have to combine it with matrigel, with gels, you have to use the round bottom, magnetizing the cells. It make it easier for you to do a lot of the downstream work that it's complicated. And usually when I mean complicated, it means it compromises reproducibility. And that's one. You want to reproduce results. And if it's not reproducible, you waste time because you have to redo it. You have to re-optimize. So the way I see magnetic 3D cell culture is an enabling tool to make things easier when you're doing 3D cell culture. And it's very simple. This is an example. You have a well and you have a magnet right on the bottom of in the center, in the bottom of the well. And when you magnetize the cells just go to the magnet and form the 3D sculptures. And the same magnetic field you can use to hold and manipulate your cultures. So because we use magnetic field, we can promote cell-cell interaction. We apply the magnetic field, the cells come together. And usually a lot of cells, when you promote cell-cell interaction, push them together, they form a 3D structure really fast. So, and it's very reproducible in size. You can always add complexity, as you see here on the top right, where we culture glioblastoma, GFP label with normal human astrocytes, Min-Cherry label. We culture them separately, and then we magnetically brought them together. And then you can see over time, once they touch each other, the glioblastoma basically overtake the culture and invade the normal human astrocyte, which is a good example of, metastatic process when the cancer cells invade the normal tissue. So, and this we published in Nature Nanotech as well. We have a really good paper in neoplasia that also we actually use patient cells and show this process in vitro. The bottom right, it's work that I always show, and it's also it's a good example of the practical power of magnetic 3D cell culture. The only reason we got this beautiful image, which was the cover of Tissue Engineering, was because we stained the tissue whole mount. What do I mean by whole mount? It's basically you have your spheroid, you put on the magnet, and you stain the whole spheroid together. You don't have to slice. You don't have to do the paraffin embedding. And that's just the way you do with the monolayer culture, right? You have your monolayer culture, you do your staining on the slide, on the plate, and when the cells are attached. But with the magnetic field, now we can do whole mount. And of course, from the biology side, this was a really cool work too, because it also goes back to the point that I keep making it, that we have to accept some complexity when you're doing 3D cell culture, and you're gonna probably have to optimize if you're looking for the right phenotype to answer your questions. And this is when we start doing this work, this is a co-culture of pre-adipocyte stem cells with endothelial cells. The red here is pure lipin, which is a marker for adipocytes. The green are GFP labeled endothelial cells. And when we start doing this work, we really wanted to see the phenotype for the endothelial cells within the culture, which was the sprouting, as you can see here, the stretching, starting making the tubular structures of vascularizing the tissue. And the reason we're doing this work, we are looking into the relationship between fat, obesity, and cancer cells. So we really wanted to replicate the endothelial phenotype. And when we started doing this culture, this work, the question was, which ratio of cells do we use, right? How many of the pre-adipocyte stem cells, how many of the endothelial cells? And we started with 50-50, right? That was the obvious point for us. And I can tell you, it worked. The cells integrated as far as mixing the cells together, the endothelial cells and the pre-adipocyte stem cells. But the endothelial cells did not sprout and form the phenotype that we wanted. So for us to get to that phenotype, we had to optimize and play with the ratio of cells. So for us to get this phenotype, we had to go 95% pre-adipocyte stem cells, 5% endothelial cells, and then we saw this sprouting. So again, to achieve the complexity, you have to optimize. So, and it was once we got these results, again, this was early on, and I like to say this was early on when we first started, because everybody has heard the name organoid, right? And we actually, on this paper, we used the term organoids. And I actually went back and looked how many papers before this publication had used the name organoids, either on the title or abstract. And there are 20 papers only that have used the term organoid. So this work, it's early on. And so but back to the biology. So when we saw that by optimizing the cell ratio, using the right cell ratio, we get the correct phenotype. We get the morphology that shows in vivo biology. So the hypothesis we had next was, what about if we get the exact ratio that is in vivo, right, of the different cell types? And what would that do? So what we did, we took a mouse, we took the fat tissue of the mouse, we removed the adipocytes, we took this whole stroma vascular fraction, all the cell types that are in there, magnetized the cells, and we reassembled, right? So it was all dispersed cells, we assembled them, and after 2 weeks, we stopped and we looked at the morphology of the tissue. And here, yes, we did use frozen sections. So this is a frozen section of a reconstructed in vitro tissue. And here, the green is CD31, which is a blood vessel marker. And on the right here, the decorin, DCN is decorin, which is the stroma marker. But if you look at the morphology, look, the blood vessel marker, CD31, have this circular morphology that looks like a blood vessel. So, and the stroma cells are in the right spot too, supporting the blood vessel formation. So the bottom line here is, if you get the right ratio of cells, the cells will do what they know how to do. They reorganize and they form these organotypic structures that really mimic in vivo biology. And of course, we published this in Tissue Engineering a few years ago. And this is a beautiful image with a group in Boston. Actually, there are two Brazilians collaborators. And they do a lot of work doing with the adipose tissue. And this is a confocal images, the Kagawa instrument that you can see the red here, the perliping stained cells, and it's a beautiful image. So again, these things are the type of images like this, doing a whole mount staining, it's not always trivial in the 3D cell culture. Okay, so as I said, we started with at MD Anderson, we moved to we spun off a small company, Nano 3D Biosciences, and as a small company, we worked really hard to publish in big journals. Best marketing from a business part of is a good publication. So we worked really hard in publishing really good journals. We published high impact journals. And the reason I mentioned that too is that we use the nanoparticles. So it's a fair question to ask, what did the nanoparticles do to the cells? And we've published over and over. It doesn't do anything. The nanoparticles we use are bioinert. Again, not all nanoparticles are made equal. Some nanoparticles are very toxic. The ones we use are not. And we have a white paper, 5-page, that shows all the results, published results, have all the references that show viability, proliferation, stem cell differentiation, protein expression, gene expression, inflammatory stress, oxidative stress, phenotype comparison, with and without the nanoparticles, and there's no difference. So, and also we did we had a big grant from National Cancer Institute, from the NIH, with the Scripps in Cold Spring Harbor, where we did, and I'll show some results here that we really, we validated our high throughput screening tools. And before we start working with them, we validated several cell lines with the control compounds, with and without the nanoparticles, and the results were exactly the same. So the nanoparticles did nothing as far as the drug interaction of the interaction between the drug and the tissue. So just to show, it's a fair question. So this is a quick movie that shows exactly workflow for magnetic 3D cell culture. So we have a kit. So from a commercial point of view, there is a kit that comes with magnets, 2 different type of magnets, comes with nanoshuttle. That's the nanoparticle. You take a nanoparticle, you add to your cells. Cells can be attached or in suspension. The nanoparticles, electrostatic interaction with the cells. You wash your sample just like you do with monolayer culture. You detach, you count, and then you're going to put your cell repellent plate on the magnet on the magnet array or the magnet drive that has a magnet right in the middle of the well. So you dispense your cells. As soon as you dispense their cells, the cells go towards the magnet. And this promotes cell-cell interaction. You need to leave it on the magnet. 15 minutes, the cells come together. You can take off the magnet. And then cell-cell interaction takes over and makes a spheroid. Now, you want to change your media. You want to add a drug. So you put on the larger magnet, and then you can pipette your media out without losing your spheroids. Add your fresh media. Then you can add your compound, and that's it. So the workflow is the same as monolayer culture, with the exception of adding in a particle and putting on the magnet to hold the cells down. And here we show we have 96, 384, 1536, but we really have all the way from 6 to 1536. So one word that I like to use is enabling tool, and that's what nanotechnology is. I'm a chemist by training. And that's why nanotechnology is here, is to help, to facilitate, to enable things that we do in the lab, want to do in the lab. And this is a good example of enabling property of magnetizing cells. And here we're using NALM6. These are leukemia cells that do not form 3D structures, but we still magnetize the cells to facilitate visualizing the cells, putting the cells in the middle, and using this tool for evaluating CAR-T targeting, okay? So the way we did this, we took NALM6 cells that were modified. One was CD19 negative. The CD19 negative cells were green. The CD19 positive cells were red. We magnetized them. We brought them together to the middle. And then we had the CAR-T that should target only the CD19 positive cells. And then after 48 hours, if you look, the CD19 positive cells went away, which means the CAR T killed the right cells. So it was just very easy to do this by just putting them in the middle of the well. Instead of having to scan the entire well, look for red and green, this was very straightforward. And we had a grant from NIH with Lawrence Cooper's group at MD Anderson to develop this tool. Sometimes you want to make several spheroids in the same well. And the way we do it with the magnetic 3D cell culture, for example, we use a 384 magnet drive on the bottom of a 96 well cell repellent plate. And what this does, you can create several spheroids in the same well. And they're fairly reproducible in size. And there's many reasons why you may want to do different several spheroids in the same well using this approach. So using the same thing, and this was work we did with Agilent using the Citation 5 and in their automation system. And here they want to use the quantified T cell killing, but they wanted to use several spheroids in the same well. So what we did, we took the 384 plate, and with a 24 well plate, a 384 drive, and we made 9 separate breast cancer spheroids in the same well. We had the CAR T, and we're able to see the killing of the cells. This is time over the confluence of the imaging, and these curves allowed us to quantify killing of the tumor cells. So again, it's enabling tool. It's not easy to do this especially, and one thing that I should emphasize, usually when you use the magnetic 3D cell culture, you can use a flat surface. So for imaging, it's very easy to do it. Just like here, there's no round bottom for 24 well format. So that's not easy to do. You can do it, period. And also for spatially distribute them in a well, if you have a round bottom, you cannot do that because they all roll down to the middle. So another tool that we keeping the enabling tool, transferring spheroids from well to well. It's not, again, another step that is not easy because people have to cut the tip of the pipette so you don't break your spheroids when you're pipetting from one plate to another. So we came up with a magnetic MagPen, which basically is a tool for you to pick up the spheroids, see your spheroids there, and transfer to a fresh plate. So it's very easy, and this is a quick cartoon movie that shows exactly how this works. So the multi-mag pen, it's a tool that you have your magnet, you have a sleeve, we call it a special sleeve, that is sterile and cell repellent, and you have a plate. So the first step, of course, you coat your cells magnetically, your cells are magnetized, but now you want to transfer for a fresh media, or you want to do immunofluorescence staining to fix your cells and move from the well you are. So you put the sleeve, then you put the magnet on, and then you can just pick up your spheroid. You do this motion just for cells to move, and you catch the magnetic field and go up to the sleeve. And then when you lift up your magnet with the sleeve, you can see the spheroid. Of course, this is a cartoon, but you saw in the previous image that it does pick it up. Then you take a magnet off. And after you take magnet off, we do put the magnet on the bottom just to gently pull the spheroid off the drive. And then you can remove your sleeve. So you put your magnet there, and you can remove your sleeve. So here we have this in 24 we have a single tool to pick up single spheroid. We have 24 and 96. So you take off the sleeve, and that's it. So it's pretty straightforward. It's not a simple task, and it's very easy, especially if you're doing immunofluorescence staining. It can be very, very helpful. So now back to, sort of the first topic. Is it 3D really similar to in vivo? And this was work done by MD Anderson. This was we use triple negative inflammatory breast cancer cells. We took the patient cells, we grew a PDX. This is the tumor. the patient-derived xenograft. And what we did, we took the tumor, split the tumor in half, and then we came up with a protocol. We break the tissue, magnetize the cells, bring the cells back together. And the question is, does it look like the original tumor? And this is the answer. So we look at Icadira, Vimentin, Ki67, Pismad. So this is the tumor on the left, and these are two different bioprinted cultures. And this is our paraffin-embedded sections that we did the immunohistochemistry. And when you look at cadherin, as I said, cadherin is very important for 3D biology. You can see that the morphology is very similar to the in vivo tissue. Then you look at Vimentin, same thing, very similar morphology. And then we go to Pismad, same thing. So we took the tissue, compared to broke it apart, reassembled, and compared to the in vivo, and it's the same. And in the same work, not only the morphology is the same, the phenotype is the same, we also did gene expression profiling, and there was no difference, which also shows that nanoparticles don't do anything, because this is the ultimate comparison, right? A pristine tissue, we took the same cells, magnetized the cells, reassembled, and looked at just about everything of the tissue that was recreated, and the biology was the same as the original tissue. So here is the work we did with metastatic prostate cancer that metastasized to the bone. We took the needle biopsy, we removed the bone chunks, magnetized the cells, reassembled, and within 96 hours, we got this beautiful dose-dependence curve to doxorubicin, which is a drug that they use to treat this type of cancer. And the value here is where, the personalized medicine aspect. Can you take a tumor from the patient, break it apart, now generate hundreds of small cultures and test several drugs and see what drug works best to the patient. And that's what we're going towards. And we can do it, and it is effective. So now going to high throughput work, high throughput tools. And this work we did with Scripps. This work was funded by NCI, as I mentioned before, and it was a collaboration between Scripps, Cold Spring Garden, and at the time was Nano 3D. I was in Nano 3D. And here is the 1536 magnet array, very dense array. But here I like to show that, you magnetize your cells, you put on the magnet, 15 minutes, the cells are in the middle of the well, where you show the cell-cell interaction. Of course, when you're doing 3D cell culture, often you want one spheroid per well, and you want a spherical spheroid. And if it's a spheroid, it's spherical, but sometimes we call it spheroids, and they're not necessarily spherical. But in this case, you can see every well has a single spheroid on a flat plate, and very reproducible in size. From a practical aspect, and that's if you've done organoid 3D cell culture on a flat plate, with time, thermal motion makes the spheroids move in the well. So, and what you see here is a 384 plate. Every dark dot here is a spheroid, okay, that you see here. But because of thermal motion, the spheroids move to the side of the well. And when they move to the side of the well, it's very hard to image. And you lose that data point. With the magnetic 3D cell culture, again, if you put it back on the magnet for 30 seconds, you can recenter all the spheroids, which make it much easier to find and to image the spheroids. So again, it's a small, but it's a big deal if you're doing high throughput imaging of cells. Remember, as a simple example, if you're using the IncuSite, I don't know if you guys are familiar, even the Citation 5, if you're imaging a 384 plate, it takes about 20 minutes to image a full plate, okay? And this is the objective, stops in one place and move on. So if you have to scan the well to find the spheroid, right, if it's not right in the middle, if you park, you have to move the objective twice. in the same well. That means a 20-minute essay, now it's 40 minutes essay. If you have to look harder, and that time just keeps increasing. So 20 minutes to 40 minutes, it's a big deal. And if you imagine, as I'm going to show here, if you're screening 150,000 compounds, that makes a big difference. So the point is, centering the spheroid, it's very important if you're doing high throughput screening. So, of course, we're talking about 3D biology here. In the image of the spheroids that we did with the work. And here are different cell types. The top is 2 cell lines, the top 2, HT29, ovarian cancer, PANC1, pancreatic cancer. And you can see you magnetize the cells, you bring the cells to the middle, they make beautiful spheroids. And in this work, we actually use patient cells. AGM1 is one patient, HT1 is a different patient, and these are patients that had pancreatic cancer. And Cold Spring Harbor isolated these cells from the patients, and they separated the cancer cells from the CAFs. The CAFs are the cancer-associated fibroblast. And if you look at this, so AGM1 and AGM1 CAF are pairs. So these are the CAF from the same patient. And same thing with the HT1. So the CAFs, the cancer-associated fibroblasts, you bring them to the middle of the well, they make beautiful spheroids. Generally, fibroblasts do that. But when you look at the cancer cells, they would not make the beautiful spheroid. They make these structures that are all dispersed. And when we look carefully at it, they're organoids, which means they have this cyst-type structure, which is particular with organoids. Yes, it can be confused with organoids, spheroids. What are the difference? Generally, organoids, they have the organotypic morphology, and they generate from stem cells. So in this case, cancer stem cells, and that's what they do. They do not like to form a cohesive structure. But there are ways to make them create a cohesive structure, and I'll show you in a few slides. So as you say, cells do what they do, but still magnetizing the cells made it easier to center the cells, manipulate, wash, and all that. So what about the results here, right? So part of the hypothesis of this work was to show that 3D cell cultures give less false positives than 2D cell culture. And what are false positives? False positives is a drug that you test and tells you, the results tells you that they should kill the cell, they should work in vivo, and when they go in vivo, they don't work, right? And part of this false positives, we do, and these results show, part of the reason is because of the 2D aspect of the culture. So what we did here, we took a compound library, 3,300 compounds, which did not have any cancer-prescribed drugs. And we tested this in 2D and 3D, all these compounds, with the patient cells, okay? So as you can see, every output of the screening of the 3,300 compounds, every screening gave you lower number of hits of cells that killed the cancer cells in 3D and 2D. So, and that's what we say as reducing the number of false positives. We had less hits of compounds that are not supposed to kill cancer cells in 3D and 2D. So that was good. It followed the hypothesis. Now you may ask, you have the same compounds, why you get different numbers of active compounds for each cell type. Remember, these are cancer patient-derived cells, and that's why the value of personalized medicine. Every patient behaves different to cells, they behave different to different drugs. So then we went a step further and we looked at the sensitivity of the cells to the drugs, and we plotted them as inhibition sensitivity on 2D, sensitivity in 3D on the y-axis, sensitivity in 2D on the x-axis. So what this means here is that every dot here is a compound, and if the dot showed sensitivity that falls within the white quadrant, that means it didn't kill the cells. Which makes sense because this is a compound library that had no compounds for cancer. So most drugs should be okay. Next, the compounds that are in the pink quadrant only killed cells in 2D. It did not kill the cells in 3D. Those are strong, false positives. So, and then on the green are the drugs that killed both, killed in 2D and 3D. So they behave the same, which fine. But what's really interesting and relevant is that basically the only drug that only killed the cells in 3D, but did not kill the cells in 2D, meaning we did the screen on 2D, the cells were fine, was disulfiram. And disulfiram was actually in phase 1 clinical trial. We didn't know. I was just happy there was an aspirin. So, but yes, so we not only reduced the number of false positives, we also picked the drug that, as we say, the needle in the haystack, right? We took the cell that was only working in vivo, didn't work in 2D, and basically validated that also that you can 3D cell culture better mimics in vivo biology. So we went on, we did 150,000 compound screening. We published this in SLAS Discovery. But if you're interested in high throughput screening, what is very important here is the Z prime, right? Z prime is the distance between the positive negative control within the assay. Basically, is the number of standard deviations that separates, and if the Z prime is higher than 0.5, it's supposed to be excellent assay, and that's what you're looking for. And constantly we have very high Z primes, which means reproducibility within with 3D cell culture. Importantly, though, this Z prime was not measured using imaging. The Z prime was using cell titer glow, which is chemiluminescence assay, which is, it does make a difference because imaging it's very hard to get this high of Z primes because it's hard to have the sensitivity on the size difference of the spheroids. Although I'm going to show some results that have excellent Z primes using an assay that we developed just in a few slides. So interestingly, right? We took the cancer from the patient, pancreatic cancer, remember? And we separated the cancer cells from the cancer-associated fibroblast. And we tested with several drugs, thousands of drugs. So after we did that, the question was, what about if we bring these cells back together? What happens, right? So, and this is what I said, there are ways that you can actually bring all cells together. And what we did here to see what happens, so we can also monitor the different cell types. We stained the CAFs, the cancer-associated fibroblasts, with a red stain, with a red cell tracker. And then we took the cancer cells and we stained with the green cell tracker, okay? And as you can see, the red cells, the fibroblasts make beautiful spheroids, the green cells make the organoids, behaved as we expected. Now, when we mix the cells together, so here you have the CAFs and the cancer cells. Look at 24 hours. All the cells come together, right? Remember, the cancer cells did not come together, but they all come together when you co-culture them. And then the tissue starts to reorganize and the cancer cells come to the outside. So cancer is very dynamic. And so time is very important. So if you do your assay at 24 hours versus 96 hours, we'll get different results. Now, and all goes back to what is your question, right? You may want to do it 24 hours. You may want to do it 96 hours, but you'll get different results. So you got to be very careful when you're doing assays, which time point you're going to do it. And if you do it after 2 weeks, it's going to be also different because these structures are going to grow very much. And then you have other type of issues like necrosis, hypoxia. So when you're doing 3D cell culture, the physics, the physical size and the biophysics of the structure is very important. You have to keep that in mind. So as I showed you, we brought the tissue together, the cells together, and you can see in 24 hours, clearly there was motion, although I'm not showing a time lapse, because you came from a bigger size to a much smaller size where all the cells are in a very small space. And this happens a lot with just about every cell type. So, and the reason we can observe this very easily is because we have a flat surface, right? So when you focus the cells in the middle, there's no gravity pushing them, so you know it's cell-cell interaction when you just push the cells together and put them in contact. So can we use this cell-cell interaction, this mobility due to cell-cell interaction between cells of the same type, different types, as a biomarker, basically, for biology? Can we detect killing? Can we detect other phenotypes change due to drug action using this mobility, right? And so what we did, we basically used, this is a good movie. Let me show you the movie. It's easier that way. So basically what we did, we took the cultures, we made the dot, we call it dot. We brought the cells in the middle of the well very fast, right? Add a drug and took off the magnet and start imaging the cells. So what you see here, every well has a spheroid. That's what you're seeing here, okay? And we use, these are 3T3 fibroblasts. We use retinoic acid, which is it's vitamin A, also is used as chemotherapy. And this is just a toxicant. So we have high concentration on the top here, low concentration. What's important is the time. It's fast. And the bars here is just changing size. So watch what happens. So 16 minutes, see the control, which is a bit counterintuitive. It's getting smaller much faster. So 3 hours, you already have your dose sensitivity for this. So because the control contracted much faster. But what is really sort of unique for this assay are these curves here that you see here. And this exactly movie is published in scientific reports. So you can get that in the publication. So what does this tell you? So you have time and you have size change, right? So the high concentration, this top, changed very little in size. Why? Because you brought the cells together. Because the cells are dying, they're not interacting very well because they're dying, so they don't move as fast. So the control, where there's no drug, move very fast because they're pristine, right? So the shape of these curves is very unique to drugs and to cell type. So you can use how the cells move to fingerprint the cell type or the drug. So here's just to show, standard traditional live dead assay where we have the control here. As you said, it contracted fast and the cells were alive. It contracted slow and the cells were dead because it shows the red. But if you look at the curves here, again, time and size change, right? So the red and black, it's flat for dexamethasone. Why? Because the cells are dead. They're just moving. We add the high concentration, they just stay there. But the shape of these curves, how different. So SDS is the positive control. You have this, which SDS is a surfactant, basically dissolves the cells, right? It breaks the cell membrane. And very different curves. So if you look at every set of curves, you look here, are representative of the drug they're doing. They're all different. So this is the same cell type, different drugs. Now, we went the other way. We used the same drug, which is doxorubicin, in different cell types. We use lung cancer, colon cancer, and liver cancer cell lines. Okay? Doxorubicin, as you can see from the blue line that barely moved, killed every cell type, right? The controls contract to every cell type. But one thing that is key, the shape of the curves, all different. So here you have the same drug, but every cell type have a different response to it. So what we did, we just did a polynomial fit for these curves, very straightforward. And then we took the cofactors of this fits, and we just did some math, and we did principal component analysis, which is a tool for, again, to simplify how we look at this data and be able to plot this three-dimensional data on a 2D plot. But what you can see here from the PCA analysis is that it's a dimension reduction approach, math approach. So liver, lung, and colon, they map very different. They cluster very different on a PCA plot, which is interesting because you basically can fingerprint them, and the concentration too is different. Now, for us to get this data, right, we use a 384 plate, taking an image of the full plate every 15 seconds, right? So remember I said, if you use an INCU site or Citation 5, it takes about probably 40 minutes to take, one single well because you're scanning for every. We took a full plate every 15 seconds. Now, instruments that do that, actually very few instruments, if any, can do that. The only reason we were able to do this, we didn't use a microscopy. And we use a microscope, we use an iPod. So because the nanoparticles give you contrast and you can make a shape, we can use as simple as a mobile device to get this real high throughput data, really sensitivity data, but just looking at cell-cell interaction. So here we're just showing that, yes, the cell mobility can be used as a very effective biomarker to look at cell interaction. And we took further. This work we did with AstraZeneca, and now the drug we did, the test we just did, we're looking at cell mobility. Now we're looking at vasoactivity, which basically we take vasoactivity. So what is vasoactivity? Vasoactivity is when you test you do for just about every drug, because in the way they do this test, they take the aorta of the rat, they chop it up, and they stretch with the drug present. And it's not very reproducible, and it's very slow. So the hypothesis we came up, can we take vascular smooth muscle and make a ring and watch this vasoactivity of the drug? So this is a movie that we show. We use blebbistatin. Blebbistatin is a vasoactive drug, and this is overnight. So we have high concentration of blebbistatin. You have a control on the bottom. And blebbistatin is a vasodilator, so it makes the cells move slower. It blocks phosphorylation myosin light chain. But look at this, at these rings here. Overnight, you can detect, and it's clear that the drug is, a vasodilator. And then we can measure those curves. We can also, and we did the staining to make sure that the biology is right. So as I mentioned, blebbistatin blocks phosphorylation myosin light chain, and that's what it did. You have your control. The red here is myosin light chain. You have high concentration of you have blebbistatin. It blocks the phosphorylation. And we tested vasodilators, vasoconstrictors. We did cell line, and we did human, and they're very different. And we published this in scientific reports with AstraZeneca a few years ago. Now, if you're interested in doing high throughput screening, I just want to show this quick movie here just to show that, and this is not trivial. High throughput screening with automation, if you're using just without the magnets, it's very hard. So here you have your magnet drive, you have a 384 plate, you put your cell repellent plate on, you have your cells back here. The robot will take the pipettes, it removes the cover of the lid of the plate. It's going to pipette your cells. We'll pipette your cells, we'll dispense your cells. The cells are magnetized. As soon as they see the cells, they'll come to the middle. And this was done by TCAN as a collaboration. We presented this, and we have an application note for this as well. You put a plate on. In this case, the workflow left in the magnet for 60 minutes, and then the magnet the robot comes, picks up from the magnet, and puts inside of the incubator. Now you want to wash, it puts back on the magnet, it washes cells. So I'll automate it, but the magnet is the key to hold them down. There we go. And again, as you see the bottom, Z prime, very high Z prime, reproducibility size with different cell number, very reproducible. So and that's what we want when you're doing 3D cell culture. This, again, back on the, still on the enabling tool, we can use imaging and we can use chemiluminescence. And in this case, AstraZeneca published this high throughput screening, but they're looking at electrophysiology in this case. They're using cardiomyocytes, look at beating. And the only reason they could do this is because they put the magnetically brought the spheroids into this electrode, and were able to get the right response. And they published this in scientific reports. They also did co-cultures here where they use fibroblast and the iPSC-induced pluripotent stem cells. So it's really good work. I'm not going to get into it. We call bioprinting, right? Everybody talks a lot about bioprinting. Again, back to the Einstein, you can make things, you know, simple as possible, but not simpler. Again, we just need a spheroid. We just need a dot. We need a ring. We can make a ring. Do we want to make it more complex? We can. We don't have to, so no need for it. So if you want to, you know, write an N here for nanoshuttle, we can do very complex structures or shapes and patterns as well using the magnets. And we're very proud of the work we did. This was a few years ago. We had a grant from CASES, and this is Peg Whitson right after she was in space using our system, creating 3D structures, manipulating 3D structures in space. So, quick. In space, there's no gravity. Cells grow in 3D. So why would we want to need magnetic 3D cell culture in space? Manipulating cells in space is very hard. So the idea here was to send the cells magnetized, use the magnet to facilitate manipulation of the cells. And we also, another goal of this work was to create the first monolayer in space. And we took the cells, we did stick the cells on tissue-treated membrane. The cells stuck, but they never spread. They still grew this way. So there's more work for us to do. There's a couple different things we can do to create the spheroid in space, but we're not successful. But it was very interesting that the cells, even though they're attached, they would not spread into the. And of course, we have lots of collaborations. We had lots of funding from NIH, from National Science Foundation, from CASES, State of Texas, lots of collaborators. And I guess we have our question answer at noon, and I'll come back in to the question answer.
Thank you, Glauco, for this wonderful presentation. We like using your nanoshuttle system.
Thank you, Leda.
Hopefully you have time to see this presentation from Lefteris.
All right, so I'll come back at noon, right?
Yeah, but if you can attend this next presentation.
I will stay on, yes.
Okay, thank you.
Thank you.
Yes. So now we're gonna present our next presenter, Dr. Eleftherios Michailidis. He's an assistant professor in the Department of Pediatrics at Emory University School of Medicine. He joined Emory University in 2021 and has established a multidisciplinary lab focusing on virus-host interaction in hepatitis B virus and other viruses. He's also a member of the Discovery and Developmental Therapeutics Research Program at Winship Cancer Institute of Emory University. He received his PhD in microbiology and immunology from University of Missouri in Columbia, Missouri. He completed his postdoc fellowship at Rockefeller University in New York City. And the Michaelides research program focuses on understanding the interaction between viruses and the host using a set of biochemical, cell-based, and in vivo methods. In particular, the group is interested in HBV and the development of eradication strategies that involve state-of-the-art primary hepatocyte systems and humanized liver chimeric mice. These systems expand the scope of the research beyond viral hepatitis to other liver-related disease and fields, including fatty liver disease and liver immune metabolism. In addition, they have a strong interest in antiviral mechanisms carried out by interferon-stimulated genes and other host proteins, but also small molecule inhibitors in regards to mechanisms of action and resistance. In this direction, their main focus has been HIV, HBV, and SARS-CoV-2. To accomplish these goals, they use medium and high throughput genetics screens, gene overexpression, and CRISPR knockout across different cell systems and in some cases in humanized mice. Thank you for participating in this workshop, Lefteris.
Hello, everyone. Thank you, Leda, for this nice introduction. And it's also very nice to follow up on the previous presentation. I think was very exciting. And we work on primary human hepatocyte systems, using them for hepatitis B virus and other liver diseases. HBV is a global health burden. There are about 300 million people that are chronically infected with this virus, even though we do have effective vaccines. Those that are already infected, the vaccines are not really doing anything for them. And we do have treatments, but even with that, about 820,000 people die each year from HBV-related complications, such as liver disease and liver cancer. And there is still ongoing transmission in resource-poor regions that don't have vaccination programs that cover everyone. So the major need in our field is to have a widespread cure that will be applied similar to other curative strategies that were very successful for hepatitis C virus. We want to see the same for hepatitis B virus. HBV is a very small DNA virus. It's a 3.2 kb genome, but it's a very complex virus in respect that it employs a DNA and an RNA genome, and it can stay very stable in infected hepatocytes. So this is a simplified version of this life cycle where the virus enters through a receptor. It was discovered about 10 years ago. It's a bile acid transporter called NTCP. And through this receptor, it enters hepatocytes, and it can deliver its genome in the nucleus, which is a partially double-stranded genome that is repaired by host polymerases to form what is called covalently closed circular DNA, or cccDNA. The cccDNA is very important because it is a DNA form of the HBV genome, and it's the template for transcription of all viral mRNAs. And it stays very stable in infected hepatocytes, and any treatments that we have approved target the viral life cycle outside of the nucleus. So the cccDNA stays stable, and even if there is a block in the cytoplasm, there is a recycling pathway that replenishes the cccDNA. So approved therapies involve pegylated interferon alpha, which is an injectable treatment, and it is quite toxic. So there are side effects. So a lot of countries now are moving away from interferon regimens, but it's still one of the approved therapies that can lead to a cure in about 10% of the people that receive it. And the other type of medication includes nucleoside analog inhibitors. These are the same drugs or the same class of drugs that have been used very successfully for HIV infections, and they are very well tolerated, but they do not cure from the virus. They block reverse transcription in the cytoplasm, and they do not really affect the cDNA unless it's a very long-term treatment. Some of the big questions and barriers to HBV eradication that we are interested in have to do with the stability and maintenance of the cccDNA in chronic HBV and under therapy. We're also interested to know how interferon alpha leads to a cure, even in a small subset of patients. And this has to do also with differences at the virus level in terms of HBV genotypes. And also we constantly work towards understanding this interplay between virus and host. So we're looking for viral and host targets for achieving a cure. To accomplish all of this, we realized early that there is a need for efficient in vitro and in vivo HBV systems because this is what has been lacking for the HBV research. In addition, as I mentioned, interferon alpha cures about 10% of treated individuals. And even though interferon-containing regimens are not a preferred treatment option, a better understanding of the mechanisms of action could be used for developing new antiviral strategies without the use of interferon itself. And the HBV pathogenesis and liver disease progression studies would benefit from better insights on the role of the HBV genotypes for interferon sensitivity, but also for liver disease, but also for mutants that arise during the course of infection. And these are isolated usually in the pre-core region of HBV. And not a lot have been has been done in order to identify what are the host determinants of these differences in HBV diversity. So in terms of our studies, we focus both on the host and the virus. For the host, we look at cell-intrinsic responses, and we look also, as Leda mentioned, at ISGs, interferon-stimulated genes, but also broadly to host restriction factors and host dependency factors. And for that, we employ a lot of genetic screens and multi-omics studies that I will talk about later. And at the virus level, we started using some approaches to better study HBV genotypes, mutants, and further we are looking at co-infections and comorbidities of HBV, and this is in combination with HIV. And being in the division that has a big focus on antivirals, we're also looking mechanistically how these novel antivirals that come into play, how they affect the virus, but also if we can trigger innate immune responses, how they can further enhance their activity. So HBV is a hepatotropic virus, which means that it infects only the liver and only human hepatocytes. This limits the systems that we can do these studies into hepatoma cells, which are cancer cell lines with the overexpression of the receptor NTCP, but also primary human hepatocytes, or as they are called, PHH. Unfortunately, mice are not permissive to HBV, and therefore in vivo models that try to understand HBV biology are not approaching this from all aspects. So there are mouse models that will be transgenic mouse models that can have a form of HBV that doesn't really fully replicate the whole life cycle. So there's no cccDNA, for example. And there are also mouse models that I will be talking about today that are humanized, and they can support HBV replication, the whole life cycle, but they lack immune system. So there is no perfect model, unfortunately, mouse model in HBV. So that's what also shows here that unfortunately we have this limitation. In the context of in vitro studies, so cell culture studies, even though the virus itself is very infectious, in vitro, very high virus titers are required for efficient infection. And it is very challenging to detect and quantify the cccDNA. Southern blot assays, which are very laborious, are typically the gold standard for this kind of detection. And even then, it's not very efficient to detect in several systems. And not only we need high virus titers, but even when we infect the cells, unlike other viral systems, there is poor virus spreading, which limits our studies of the complete viral replication cycle. And as I said, we can use also hepatoma cells like HepG2 or HUH7 with the addition of the receptor NTCP. But these are cancer cell lines that have impaired innate immune functions and cannot fully recapitulate host responses, at least host intrinsic responses. So primary human hepatocytes or PHH are the gold standard for HBV-related studies. Those come also with their own challenges and limitations because they are isolated from deceased individuals. There is donor-to-donor variability and they're not renewable. They are terminally differentiated cells that do not proliferate in culture. They are very expensive to obtain and they cannot be modified genetically. And one of the drawbacks of the PHH is that usually they come cryopreserved, and then when you thaw them, you lose a lot of the cells. And even if it's a good batch of cells, once you plate them, they start deteriorating in culture after a few days or 2 weeks, and their functions are not as robust. And also for this kind of systems, we still need to use high virus titers for efficient HBV infection. So the solution to this problem, at least for us, was to use a mouse model called the FAH knockout. This is a liver injury mouse model that is immunodeficient. So it's an NRG background. And the idea is that we can transplant human hepatocytes into these animals. And then due to ongoing liver injury, we can have the human hepatocytes engraft in these animals, and after about 3 months, they repopulate the mouse liver. This is a slice of the liver of a highly humanized mouse, and in brown staining is the human FAH staining, and because these are FAH knockout mice, so it shows that almost the whole liver now is repopulated by human hepatocytes. And the way we have been using this mouse model is with multiple applications. So I will say first that we use this as a factory to produce more PHHs. So we start typically with 500,000 cells that we transplant into mice. And as I said, after 3 months, these animals become fully humanized. And we apply an in situ perfusion that allows for the isolation of the cells from the freshly isolating the cells from the mouse. Then we follow a Perkel centrifugation protocol that purifies the cells so we can enrich for the human hepatocytes, but also remove any small cells that are not hepatocytes in the cell suspension. What we end up having is a mouse passage MPPH or MPPHH. And this figure shows like a representative figure after culturing the cells in 2D after 1 week or after 8 weeks in culture. So we can keep this for a very long time. And the advantage is that we get also very large quantities that allow us to do unique applications. So from each humanized mouse liver, we can isolate about 100 million cells. And that's a 200-fold expansion from the initial transplantation. And these MPPHH can be further passaged for a few more passages, not unlimited, that allows us to have huge quantities of cells. So after 2 to 3 passages, we could have quantities that would be equal to 1 human liver. So in terms of HBV, because this is our main focus here, we tested this system, and we found that they can be very permissive to HBV infection and spread. This is an experiment where we infect the cells, and every week we had one plate that we fixed, and we also collected the supernatant. And as you can see here on the left panel, when we look at the e antigen secretion, one of the viral antigens, after 1 week versus 4 weeks, there is an increase in the secretion of this antigen. And then depending on the low or high inoculum, we can have more or less infection. And it's interesting that when we look at the core antigen staining, which will be indicative of what cells are infected, we see that there is an increase in the number of infected cells as time progresses, suggesting that there is virus spreading. But again, to achieve these images and these results, we had to infect the cells with high titers of inoculum. And also we can accomplish this typically with 1 or 2 viral sequences that the HBV field commonly uses. These are stable cell lines that produce HBV in the supernatant that we concentrate, and then we can infect the cells. So this limits us in what kind of HBV sequences we can test. So even though the limitations that I mentioned earlier, some of them have been addressed, so we can have virus spreading, we use PHH and not cell lines like HepG2 cells for our studies, we still have to infect with high titers, and it is still challenging to detect the cccDNA. To overcome this, I'm going to show you now another application of the humanized mouse model, where not only we can isolate cells, but we can infect the mice before the isolation. And this allows for the virus to spread very efficiently in the humanized mouse liver, and then all of the cells basically become infected. Then we apply also the in situ perfusion protocol, and we have what we call HBV MPPHH cells. And these cells maintain the properties of the regular MPPHH. So in terms of HBV, when we look at different readouts, such as secreted e antigen or surface antigen, these remain stable for at least 1 month. And here also HBV DNA. But also when we stain the cells after culturing them, we see that all of the cells express core antigen, suggesting that almost all of the cells are infected. And basically, we overcome the need to infect the cells in culture with high titer inocula. More importantly, at least for our cure efforts, we have high levels of cccDNA. This is a southern blot showing different systems, cell culture systems in HBV, either a stable cell line that produces HBV, or the hepatoma cells with and without HBV, or the regular MPPHH after in vitro de novo infection, versus the cells that are isolated from HBV-infected mice. And the cccDNA is the highest in this case. So with this approach, now we overcame the need to infect with high titer inocula, and also the challenging problems with detection and quantification of the cccDNA. So this has become now a platform for drug screens or host factor screens. This is just a proof of principle where we used this system to titrate the effect of interferon alpha and lambda 3 against HBV replication. So we use different concentrations of these cytokines, and then we plot the e antigen levels, and this can give us also the EC50s of these compounds. But also what was interesting is that even though these cytokines have a dramatic impact on viral antigens and actually HBV DNA, the cccDNA itself was not affected. This is a southern blot showing after treatment of 1 month, the untreated versus the treated samples with high concentrations of interferon alpha or lambda 3, they were unaffected, suggesting that these cytokines do not degrade the cccDNA, at least in our system, but they probably cause a transcriptional effect, and that's why we see a reduction in viral antigens. And this also is confirmed when we do a rebound assay. So in this case, we have treated the cells with interferon for 8 days, and we see a reduction with both interferon alpha and lambda 3. But then when we remove the drugs from the supernatant and we continue measuring the E antigen levels in the supernatant, we see a rebound. And what I'm showing you in the yellow line is what we want to achieve. This is what we're working on now, is to identify compounds and host factors that will maintain this phenotype even post-treatment. As I mentioned earlier, from each mouse, we get about 100 million cells. So this abundance of cells allowed us to think outside of the box and start up applications that I will present in the next few slides. So one of the first things we did was to manipulate the cells in culture by using a lentivirus-based delivery system. So we deliver with these lentis different genes. In this case, as a proof of principle, we used a red fluorescent protein that also has a puromycin resistance gene. And after doing this in 2D cultures, then we take the cells, detach them, and put them in new mice. And this allowed us to create humanized mice with modified human grafts. In this case, when we harvest these livers, and we can see here that we created basically a red liver, and then each island that you see here represents the expansion of one single hepatocyte that was initially engrafted. This will be also important when we start multiplexing with different modifications that we do in the PHH in culture and then transplant them into humanized mice. This was just a proof of principle, and since then, we have applied this methodology for HBV research, but also for fatty liver. And I'm not presenting any fatty liver related work today, but we have a paper in Cell Reports where we modified the cells by overexpressing a gene that is linked to fatty liver disease called PNPLA3. And we overexpressed 2 different SNPs, one that predisposes people to fatty liver progression and one that is not. And we have done these studies both in vivo and in vitro. So this is basically an approach that we want to take for validating hits that we find in our cell culture-based systems in a more relevant in vivo model. And apart from using lentivirus to overexpress a gene, we can also start knocking out genes. This is not very easy to do in a primary cell type because the cells do not proliferate, and you cannot easily do these studies and select for, let's say, a cell clone that has received CRISPR and has been knocked out efficiently. So everything has to be done in the dish, and then all the functional assays have to be done in the same dish that you do usually the manipulation. And this requires that the cells stay alive and healthy for several weeks. So the MPPHH gave us this possibility. And we use different approaches. One approach is the RNP delivery with liposuction. So we can deliver in these MPPHH cultures both Cas9 as a recombinant protein and the guide RNAs. So this is done with liposuction, and then we can look at the gene editing in the MPPHH. Typically, if we want to do a Western, for example, we have to wait at least 1 week because there are still proteins that need to be turned over before we see the effect from the gene editing to the protein level. And that's what you see here in a preliminary study we did targeting a metabolic gene called CYP3A4 in hepatocytes compared to the non-targeting control in black. And you see in the beginning, when we measure the activity of these cells, we don't really see a difference. But as the protein in the knocked out cells is turned over, we start seeing a decrease that eventually goes even lower. We don't have 100% knockout, and this is also dependent on the transfection efficiency that we have optimized, but also depends on the guides that you use. So another approach that we have been using that is maybe more efficient in terms of delivery, because transfection can be toxic, that we use in the first approach. We use a lentivirus-based delivery approach. In this case, we can deliver either both the Cas9 and the guide with one lentivirus, or we can have two different lentiviruses, one with the Cas9 and one where we have multiple guides. And with this approach, we have already established a protocol to edit and knock out NTCP, which is the receptor of HBV, as you see in this Western blot. And we have moved this further actually to in vivo studies where we can have NTCP knockout mice. But another important application for this, because we're very interested in virus-host interactions, is that we can use this platform for medium throughput arrayed screens. So the arrayed screen has the advantage that you can test each individual gene knockout in the context of primary hepatocytes and HBV infection. So what we did here, we obtained a library of 80 pooled guides that target individual genes, and then we deliver this as an RNP complex in MPPH8. After waiting for about a week, we infect the cells, and then after about 2 weeks, we can test with different readouts the effect of each individual gene knockout. And this is just a preliminary result, which allowed us to identify both host restriction and host dependency factors within a small screen. But as I mentioned earlier, we have already optimized our protocol to knock out NTCP, which is the receptor of HBV. So again, as a proof of principle, we want to see if this can hold also in vivo. So after delivering the Cas9 and the NTCP in MPPHH, we have editing in MPPHH cultures, and then we detach these cells and transplant them into new mice that are not humanized. And after about 3 months, we wait until the mice become humanized. And I didn't mention this, but we have very efficient ways to tell the humanization of the animal by looking at serum albumin levels, which is a very non-invasive method. Once a mice became humanized, we infected them with HBV. And then we found that, and this is the viremia in the mice, that the uninfected, the mice that were wild type, they became infected. And those that were made with the protocol that I mentioned above with NTCP knockout remain uninfected. Eventually, some of them broke through. So we're trying to see if we can make this as 100% knockout. That's why I say here, this is a partial NTCP knockout. But this allows us now to make knockout mice with this approach. A knockout only in the liver, in the human hepatocyte compartment. And as we mentioned in the beginning, there are limitations in the context of HBV sequences that we use. So over the last few years, we have been trying to address this by using also our in vivo model. So we have managed now to generate different sequences of HBV, inject them into mice, and then basically launch these HBV sequences in humanized mice. Once this happens, then we can take the mouse serum and infect new mice, store this serum, and then that's very efficient then to keep infecting mice with different genotypes and different sequences. So that will allow us to also look at hepatocyte responses to HBV infection of different genotypes and mutants, but also drug susceptibility and hepatocyte responses to different antivirals, and not just isolated on one genotype. So to that respect, I will share with you a few data that we generated in my lab, where we used genotype A, wild type HBV, and also a pre-core mutant isogenic of genotype A. And we took cells from these two different mice, and also in comparison with uninfected cells, we plated these cells. So these are basically three different types of cells. And then having them in 2D cultures, we treated them with interferon or not. And then we looked with multi-omics analysis, which includes both proteomics and phosphoproteomics. What are the genes and proteins that are affected by HBV infection and also by treatment? And based on this, and this is just a workflow, we have now narrowed down hits that we're validating using the arrayed CRISPR screening platform that I mentioned earlier. So specifically to go to some of these data, if we look at this heat map, we have triplicates for each condition. These are uninfected, pre-core mutant, and wild type. And we can appreciate that in some cases there are genes that or proteins that are downregulated, like here in green, only in the pre-core mutant cells, or they're upregulated only in wild type, or they're upregulated only in pre-core mutant. So we have already identified more than 2,000 proteins in these three clusters. So we're now doing follow-up studies in other genotypes to see which of these proteins converge and which are genotype specific. But this is still a lot to assay. So right now we are focusing on 42 of these genes because we have also done this experiment, as I mentioned, in the presence and absence of interferon alpha. So in this respect, we have these are uninfected cells, the control, not treated. And then when we treat with interferon, we basically have ISG. So these are interferon stimulated gene products. And then in the context of wild type HBV or pre-core mutant, we have proteins that, even though they behave as ISGs in the uninfected, they behave differently in wild type or pre-core mutant. So for that, we have identified again the three clusters that are upregulated only in pre-core without interferon, or they're interferon alpha dependent in untreated cells and PCM, but not in wild type. And then the third cluster where we have a loss of interferon alpha dependency upon infection. So this proteomics analysis now has led to this downstream arrayed CRISPR validation screen, which we did in an orthogonal approach, either with synthetic guides or with lentivirus-based approach. And this allows us also to add different conditions. So we do this either in the absence of interferon to see its knockout, what effect it has on HBV biology, but also with a pre-treatment and post-treatment of interferon alpha to see what is its knockout effect on interferon alpha's activity against HBV. And based on this, we have now identified host restriction and host dependency factors that we are currently characterizing their mechanism of action. So with that, I will summarize that the HBV MPPHH is a robust cell culture model for identifying host targeting antivirals. It supports studies on HBV genotypes and mutants, and the systems biology approaches provide powerful tools to determine differences between uninfected and infected hepatocytes and between wild type and mutant HBV. And the HBV downregulated a subset of interferon-induced proteins that we are now currently characterizing based on the targeted arrayed screens. And before ending, I also want to share some new data where we multiplex now the host factor screens in vivo. So each color here represents, it's an island of single cell hepatocytes engrafted initially and then expanded in the humanized mouse liver. And then we can envision that we can test different genes and different host factors, either gene knockouts within the same mouse. And we have started doing nanostring special transcriptomics, looking for the specific islands and what is their gene expression levels. And you can do this having both the gene of interest and the control within the same mouse. And to connect also with the previous presentation, we're also using our MPPH8 system to create 3D spheroids for long-term HBV infection and drug studies. This is very important because the 2D cultures, as the previous speaker mentioned, has a lot of drawbacks, and we think that the 3D cultures can overcome some of these problems. This is an image of different size of spheroids that we have generated. And what is unique about this system is that we make any modifications we want in 2D, for example, gene overexpression or CRISPR knockouts, and then we detach the cells and we form the spheroids. And this allows for a uniformal expression or modification of the cells, and not just in the surface, in the outside layer of the spheroids. And then we can study basically what is the impact of a gene knockout within the whole spheroid. But we're also interested in drug-related studies. So we use these spheroids that took us a while to make. It takes us a while to make. We don't use the magnetic approach yet, but once they are formed, we can follow them for several weeks. This has been extended now to over 3 months. And we used the capsid assembly modulator, one of the classes of investigational HBV drugs. And we show here that after treatment of about a month, we have a reduction, almost complete reduction of surface antigen in these spheroids when we look at the supernatant. And then once we remove the drug, we start seeing a rebound over a period of several months. So this we envision this as a long-term drug-related platform. And with that, I want to thank my lab at Emory and also our collaborators in US, especially Ipe de Jong at Weill Cornell, but also Kosuke Ogata from Kyoto University and Adriana Guzzi from University of Zurich. So thank you for the opportunity to speak to you today.
Vamos chamar então os apresentadores e debater. Let's call everybody to initiate the questions, okay? People. So,
Eu acho que você pode sentar aqui na frente.
any question? Oh, I know that you have many questions.
To start with Glauco, yesterday we talked about some of the presenters, and also today also you presented that some organoids could be formed in the shape of ring. And then I asked Natalia if we can customize the magnet so we can have different shapes of organoids. Is it possible?
So it's some shapes are possible. Let's put this way. Depends on the size and how much detail you need. In principle, yes, but magnetic field it's not always intuitive, especially when we start putting magnets one next to the other. They create some very unique shapes. So we can manipulate the cells with that. And also the scale is very important. What is the size that you need, right? So are you looking to make a small ring, or you're looking to make, you know, a square. And also we started working with there are tools that can make microarrays of magnets that it worked pretty well. So in principle, yes, but there are limitations for that, depending on the size.
Is there any questions? Tatiana, você consegue monitorar? Você tem alguma pergunta? Não. Okay. And then, so if no questions, I have my questions. Be selfish, but today before Lefteris presented, when this workshop was done because André Barateiro is coming to our group, and we would like to help him in his postdoc. And André presented today the work he observed in December using a monkey liver. That's a lot of material. But we are already preparing him because when he comes here to Brazil, the material will not be available in quantity as you saw in December. And we are worried because the amount is, in fact, very small than maybe André is expecting. But today, when Lefteris presented his work, we saw the humanized mouse primary hepatocytes. And I was talking to Flávio Galvão, maybe this would be a solution. So maybe we can amplify the number of primary hepatocytes instead of using mouse that we cannot use afterwards. We can use pig because pig is very similar to the human being. So
That's right.
yes, today it was a very good presentation because we can sum up we can see different approaches, but to solve André's problem. I hope André saw the Lefteris presentation, then maybe you can talk to Lefteris and then we can do some similar work here in Brazil, but instead of using mice, we can use pig because Flávio Galvão has a lot of pigs. Every week he has a pig to be used.
Some research in swine, yes. Pigs is a good model.
But just to add here, the pigs have to be liver injury pigs, right? There are actually published papers for these FAH knockout pigs.
Can I make a follow-up question to Dr. Michaelidis? Can I?
Yes.
Yeah. I was actually a bit surprised of, so like as a follow-up from the point that Dr. Suzani raised, because you really make that nice duality of expansive phenotypes that you have in culture that PHH do not multiply. Actually, you try to keep them undifferentiated for them to remain hepatocytes as they are, but then when you transplant them to a mouse, they start to multiply. So my question was more towards if there is if people know which factors trigger that multiplication phenotype within a mouse.
Yeah, so in culture, we use DMSO to help prevent the differentiation. When we transplant, as I said, 500,000 or 100,000 cells, only a small fraction of these cells engraft, maybe 1%. So there is a small fraction of cells that have this ability to engraft, either because of the space or their ability to engraft. And then those that engraft then proliferate and make these islands. We haven't published this data, but we have barcoded the cells that we put in the mice, and maybe about 1,000 to 10,000 cells maximum engraft and then proliferate. So it's not all of the cells that will do that.
So it's a donor-specific thing, like a
Uh,
cell-specific trait, or so it's
a
not environmental?
Probably both cell-specific and the area that they are engrafted. We have tried more than 15 donors, and some donors engraft better than others. There are some ways to push bad donors to also engraft, and we use some chemicals that we give to the mice before transplantation. All these are already reported in our publication from 2020. But to make a long story short, not all of the cells will engraft and proliferate.
I have a quick question, follow up on that. So do you think too that this low yield of the engrafted cells, it can be just cells will just move away, disperse? And one thing that back to the magnetic 3D, we have a paper actually that we had it was an interesting project because there was this group in Houston that had made this stem cell to address nerve regeneration for erectile dysfunction. And the problem they have, they could not keep the cells on the rat's penis. They just, and basically the way we made it work was to glue a magnet to the rat's penis. And we published that. So I'm saying there are ways that you can, and we've done that with even whenever you inject cells to make a xenograft, the cells go away. So there are tricks that we've done that work pretty well.
I mean, that could be a solution. I don't know in the liver how it would work. Maybe
because it's far. That's the tricky part. It's far.
Yeah.
But
so we usually inject them in the spleen, and then they travel in the portal vein, and then they go to the liver. A lot of them will flashed out, I guess, as you said, but some of them will find a niche. But if we put like 1,000,000 cells, the mice will die. So there is a threshold also of how many cells we can inject. But maybe by having, if the cells were very valuable, then I can see that you could minimize how many cells you put, or if you barcode the cells and you want to multiplex every time more consistently, the approach that you mentioned would be very useful.
Yeah, and we have done this with we inject the via tail vein, and we put a magnet on the side, and we did MRI before and after. And there's the other side too, that there's no magnet. And again, this is close to the skin, and the cells definitely localize where the magnet is after even the inject. So if there is a position outside that it can localize, that will sort of create a magnetic field like that right above the liver, then you may be able to use, an order of magnitude less cells and be able to enrich them there.
Very interesting.
Yeah. Yeah, and this work with it's published, with the rat's penis. I joke, if that became some kind of treatment, nobody would ever care about what the nanoparticles do, but it's okay.
Thank you. I have Kátia Luciano Pereira Moraes has a question. Is nano shuttle compatible with flow cytometry? For example, for the evaluation of immune infiltrate in the dissociative organoids.
Yeah, we've done flow cytometry work. All our users, collaborators have done it. It doesn't interfere with flow. The challenge is just the challenge as all 3D cell culture. We disperse the tissue and have a very well dispersed culture, and that's a challenge, for all tools as far as 3D cell culture. Even more challenging if they use matrix gel or something like that, that you can remove the gel on top of it. But yeah, we have data that it's very compatible.
It's
Well, if the amount of primary hepatocyte cells that engraft is so small, clearly it seems like the progenitor cells are the ones which engraft. So maybe by pre-selecting these progenitor cells, you may have a better system. What about that? What do you think?
I mean, the cell number is not an issue anymore. Like having 500,000 versus 50,000 is not an issue. Knowing what engrafts is important just for the biology of it, but not about the abundance. That's what I think. So from a biological perspective, it would be nice to know which cells, what properties they have as a regenerative type of research. But for the purpose of making humanized mice for HBV research, this doesn't really have any impact so far. But we are interested in looking what cells engraft. Yeah.
Thank you.
And I have a question for Marluce. When she presented, I was surprised that the first patient was in 2002 who received the encapsulated, not the encapsulation, but the transplant of pancreas. But my question is, now that you have the encapsulated, where are you planning to transplant, or how are you planning to do it in humans? And if there is any study progressing to humans, with alginate, for example?
Yeah, thank you for the question. Now, when we are doing these experiments in pigs, we are planning to do in abdominal cavity, and because we did this before in mouse, but in human, we have to discuss where we can put the islets with the microcapsules, maybe in peritoneum, maybe in the, how can I say, omentum?
Yeah,
Omentum. We have to study these sites, but in pigs, we are planning to do in abdominal cavity.
There are some clinical studies already using encapsulated islets. There are some clinical studies, and there's at least one company, two companies doing this, encapsulating the islets with the biopolymers and then testing them in human subjects. This is ongoing. I think it's in phase 2.
Is that?
I have a question for Lefteris. What about the off targets when you do the CRISPR? Do you see anything like that?
Yeah, so we haven't been looking at off targets like in specific genes or anything like that. But because this is arrayed screening, if there is any impact on cell health, we see that. And we don't just do like an MTT assay or something like that. We look also at human albumin levels in the culture supernatant, which is indicative also of the differentiation state of the hepatocytes.
Thank you. And I have a question for Marluce. So you use the agrons to make the sphere organoids or spheroids?
Yeah.
So when you encapsulate that, do you encapsulate with the agrons?
No.
No. You remove the agrons.
Yes,
Is it
yes.
easy to remove the agrons?
Yeah,
Okay.
They don't attach
They
in
don't
the
attach.
agar as well. So
Uh-huh.
it's
So when you make with that small device that you guys invented.
The stamp?
Yeah, the stamp.
yeah, we remove the stamp and we have the micro wells formed with agarose, the jellyfy agarose. So we put the cells on the top of it, and through gravity.
So the agar stays there.
Yes.
Oh, okay.
Yeah.
And you remove the
The cells.
the
When
cells.
you flush the cells with the media, the 3D structures come out.
Come out.
And
Okay.
that's it.
It's easy to
Yes,
remove
yeah.
them.
They don't attach.
They don't attach
No.
to the very cool system. Thank you.
Is there any other question?
I have just a question for Marlucy as well. Can
Yes.
It's
Yes.
a very simple. It's a very simple one. I got quite interested in the stamp that you used. Like, yeah, very simple question because I don't know what that stamp was made of. I understood it was made from plastic, maybe. Is that so?
Yeah.
So
What's the name of the polymer?
Polyurethane. Polyurethane.
polyurethane.
Yes.
Yeah.
So like, like I don't know who you contacted
Yes.
to. Who did you contact to do that?
Is it 3D printer? The stand.
Oh, well, you have it there? That interests me because of all that micro featured manipulation of wells. Was like some kind of technology that I didn't know back then, that if you existed in Brazil, in São Paulo or not, I didn't know who to contact. So I was a bit interested in that because, like, just to come back to that point that Dr. Suzani raised about like the yield of hepatocytes that we get from a liver, like to be very scarce. If we decrease the areas of the assays, we can get more done with less hepatocytes. So every time of manipulation of wells that we could do could be very advantage to the implementation of such models. So yeah, would like to hear from you on that.
Real quick, just to translate.
Eu posso perguntar em português, mas é de Portugal, é meio esquisito, né?
Eu esqueci disso, é verdade.
Pronto.
No, but for left daddies, I think that would be nice.
Sorry.
So he came upon the project of bettering the stamp in college. He studies polymers. They had the model, which was the original stamp, and they implemented at polymer. The course is called Polymer. It's a graduate course, and a professor there works with 3D printing, and my colleague's idea was to improve on it based on the project there. Originally, it wasn't 3D printed, right, Marlucy? But it was 3D printed originally. All right, then it was further improved through 3D printing, and several prototypes were worked on. Basically, from my colleague's initiative. Right.
You can print other types of plastics. Like polystyrene, like in plates. It's not that trivial. I have no idea.
We try another polymers, but it doesn't work. And this is the best polymer that we choose because they are not cytotoxic for the cells, and we are able to do the perfect well, so the cells can aggregate perfectly.
Printing resolution is one of the most coveted characteristics. So you can make the micro well, the the positive, right? The needles, we figured that the shape of the tips could improve the sphericity of the spheroids generated. And so we actually worked on the needles themselves, their conicity, which is basically how cone-shaped they are. That also helped when removing the stamp. But yes, basically it's empirical and trial and error in many aspects. And basically it's modeling the 3D printing model itself and resolution.
Okay, great. Thank you very much. I might get in touch.
But when you arrived in Brazil, they are from Professor Sogueiar's lab, right? So you can easily, just in the same, very easily contact them, and then maybe you can design and try and error the stamp that you would like to use. The objective of this workshop is also to do a network. So it's working very, very. And is there any other questions? Then I would like to finish, just thank you all the participants that incentivize us to do this workshop, the lecturers that reserve time to prepare the lecture and to stay here with us, the coordinators that helped us to keep this workshop alive, Professor Leda, who came from Atlanta to stay here, and the people from the computers and the transmission that helped us, and especially to Tatiane de Barrios, because without her, I could not be doing all this workshop. Thank you very much, Dr. Glauco, Lefteris, André, for being here today, and you all for being here with us in this workshop. Thank you very much.
Thank you. Thank you.
Obrigado, obrigado, Susana.
Vamos. Muito legal. Olha que a gente pode falar português. Eu acho que vai aparecer um monte de gente no laboratório de vocês, atrás do stand. É.
Querida, muito bom, muito obrigado.
Obrigada, Flávia.
Eu fico com o paciente.
Tá bom. E o Lucas, o que aconteceu?